everyone, and welcome to the Science of Childhood Cancer lecture series, hosted by St. Jude Comprehensive Cancer Center. My name is Steve Gottschalk, and I'm the chair of the Department of Bone Marrow Transplantation and Cellular Therapy at St. Jude, and together with Doug Green, coordinate the immunotherapy efforts at our institution. It is my great pleasure today to introduce Dr. Pooley to you. Dr. Pooley is a distinguished scientist entrepreneur who received his medical degree from the University of College Dublin in 1995. He went on to receive training in internal medicine and hematology oncology before moving to the Center for Cell and Gene Therapy at Baylor College of Medicine in Houston. And during his time in Houston, I was fortunate to work together with him. At the center, he focused his research on genetic engineering of T cells, and he really made seminal contribution to the field, and also was the first to translate a GD2 CAR T cell therapy study for children with neuroblastoma into the clinic. In 2005, he joined the University College of London, and since then really had nothing but a remarkable career. He has devised innovative genetic modification strategies to improve T-cell therapies and has translated his approaches into the clinic. In 2015, he founded Autolus, one of the most successful T-cell therapy companies in Europe, which currently employs more than 300 employees. Martin received numerous awards, including the Biotechnology and Biological Science Research Council Innovator Award in 2016. And according to the European Patent Office, is the most prolific CAR T-cell therapy inventor in Europe. So please extend a warm welcome to Martin, whose talk is entitled CAR T-cell Therapies for Childhood Cancer, the University College London Experience.
Thanks, Steve. Thanks very much for the invitation. So I'm Martin Pooley. I'm the director of the CAR T-cell program at University College London. So just a few disclaimers. This is the public company disclaimer because Autolus is now a public company. And then this is my usual disclaimer. But actually, my most important disclaimer is I'm not actually a pediatrician. My main interest is looking after adults with relapsed refractory lymphoma. So I'm a little bit of an interloper here. So if I sneak in some adult hematology data, please forgive me. All right. So I'm really going to split my talk up into 2/3, 1/3. The first 2/3 are really going to be about targeting CD19 and CD22 and BALL, which has been a long-term project of ours at UCL. And then after that, I'm going to talk a little bit about GD2 CAR T-cell therapy and the state of the art at UCL and what we're doing next with it. So I hope you find this interesting. So, in the UK, CD19 CAR T-cell therapy has been standard of care for relapsed refractory pediatric BALL for the last 2 or 3 years. So it's fully, as you would say in the States, reimbursed, by the NHS. So patients get it for free. And, but there are two main challenges in terms of improving long-term outcomes. And I think, that is preventing CD19 negative escape and improving long-term persistence of CAR T cells. So I think those are the two main challenges. This is our own very, very small study that we did a while ago, the CARPAL study, which I'm going to get back to you. And I think what this study shows is that if you manage to get CAR T cells to engraft well, if you kind of try and solve that problem quite well, the biggest problem you have in terms of relapse is CD19 negative escape. And most of you will be very familiar with this. This is a genomic level disruption of the CD19 gene, usually coupled with some loss of heterozygosity on the other allele. So how do you solve this problem? And I think you go back really to the history of medicine of oncology, where you target more than one thing at the same time. And the obvious thing to try and target at the same time is another early B cell antigen. And there's really only one, I think, that you can really target in this setting. That's CD22, which is expressed a little bit later than CD19, but is expressed all the way through B cell ontogeny. CD22 is actually quite a tricky antigen to target, and I think you'll get a sense of this as I go through my presentation. There's two things that make it tricky. First, it's very bulky, so it's a very big, heavily glycosylated molecule, and that prevents formation of an effective immune synapse. So you need to get T cell and target cell really close together to get good signaling. And the second thing is expressed at a lower density. So CD19 is tens of thousands of copies, CD22 is thousands or even sometimes less than 1,000 copies. So it is a challenging target. So that's CD22. How do we then target CD19 and CD22 at the same time? And there's been a number of different approaches. The simplest thing is to make two products, a 19 and a 22 CAR. That's expensive and complicated. Another approach is to try and make a CAR that sees two antigens at the same time, a so-called tank CAR, by sticking two single chains or two single domain binders together. I think that can work, but I think when you've got two such different antigens, I think that can actually be quite tricky to do properly. So we've opted in the two programs I'll discuss for co-expression, for having a T cell express a CD19 CAR at the same time as a CD22 CAR, and this lets you sort of tailor the two receptors together. And so I'm going to start off talking about the AutoTree program, and this was sort of a complete redesign program that we set up about 5 years ago now, and we kind of looked at all the different problems that everybody was having, particularly with manufacturing, vector supply, and CD19 negative escape, and we tried to sort of design our way around it. So we made a few design decisions, which may or may not have been right at the time. So one of the most important design decisions we did was to switch to gamma retroviral vectors, because you can make these vectors very easily with a stable producer. The second thing is we made this into a bicistronic vector. We have a CD19 CAR and a CD22 CAR co-expressed with a self-cleaving 2A peptide. So it's a single vector that expresses two receptors. We spent a lot of time optimizing the CD22 CAR and ended up having quite an exotic spacer, which was a pentameric motif from the comp from the comp protein, from the cartilage protein. And as you can see in the flow data, you get this really nice co-expression of the two CARs with a 2A peptide. And then the manufacturing process was a semi-automated closed system, one of the Miltenyi prodigy, as simple a process as we could possibly make, which really entailed just activation with a soluble activation media, transduction with retroviral vector in suspension with vector fusion, a brief expansion in IL-7 and IL-15, and then cryopreservation. And we've recently just published this in Nature Medicine, Cordoba et al., if anybody wants to look at this in a little bit more detail. In vitro and in vivo, when we compared AutoTree with an FMC63 CAR described by Dario Campana, which is probably very similar or identical to Kymriah, actually we had quite good performance. I think in the bottom I've put some stress tests where we've challenged on the left, where we've done killing assays at very low E to T ratios, and it seemed to outperform the Campana CAR. And then we've done some stress models in vivo, again, with limited number of CAR T cells in the face of quite a heavy burden of NALM6 cells. And again, we saw pretty decent performance, in fact better than the Campana CAR. And obviously it worked well in CD19 negative escape. The clinical study that we did in children was pretty straightforward with flu-cy conditioning. We did do it was a dose escalation, and we did split the dose in patients with high disease burden. And the patients on study really were pretty much what you'd expect, with about half of them having had a previous allogeneic bone marrow transplantation, and a reasonable mix of patients in terms of disease burden. The first thing that was actually surprising about this clinical study, and what was that we didn't see very much toxicity actually, which was really surprising based on the potency that we saw in the preclinical data. And with that, we saw relatively modest cytokine release. The clinical outcomes, I would say, were disappointing, I think, with this study. Most of the patients did go into a molecular remission. However, we didn't see the sustained response rates that we were really hoping to see. So we were hoping to see something that would give us a higher long-term molecular remission, than Kymriah, and that's not really something that we saw in this study. And I think the main reason was that engraftment was actually poor or variable. So many of the patients only had relatively short-term engraftment. And I think this is probably down to the vector usage in the manufacturing process. We did manage to learn something from this study, however. I think here's quite a complicated slide. And again, you can have a look at it in our paper if you want to look at it in more detail. What I've tried to do is sort of try and put all the pertinent information about each patient to try and figure out as much as we can from them. So we have the antigen density for CD19 and CD22 at baseline, and then the antigen density at relapse, and also the bone marrow B cell percentage and marking, or how much CAR T cells we could detect in peripheral blood by PCR. The B cell percentage in the bone marrow is a very good functional measure of CAR T persistence. I think there were some things that we could learn from this. For example, patient 7 went in with 2 clones, with a CD19 negative and a CD19 positive clone. This patient had prior CD19 directed therapy, and this patient did go into remission. And the interesting thing is that when this patient relapsed, they actually had double negative disease. So they had relapse with CD19 negative and CD22 negative disease. There were 2 other patients which possibly were informative. So patient 11 went in with double positive disease and then relapsed with still plenty of CAR T cells in his bone marrow, and also absent B cells, so functional persistence. But the relapse showed a downregulation of CD22 as well as escape with CD19, so which I think is interesting. Patient 17 was a little bit different. Also went in with double positive disease and then relapsed with CD19 negative and not, maybe even an increase in CD22 expression. But I think this patient was different in that they had started to lose their CAR T cell engraftment at this stage and had some recovery of normal B cells. And I think what this tells us is that I think going in sequentially with CD19 and CD22 CAR T cell therapy isn't really going to work that well, because you're going to end up with double negative relapses, I think, as patient 7 showed. And I think, and it's something really that's been published very nicely by Terry Fry et al., that CD22 can down modulate. And you can end up, and it's not a well characterized phenomenon, but after CAR T cell therapy, you can see CD22 dropping below or around 1,000 copies per cell in an ill-defined phenomenon, and possibly, unlike genomic disruption, it may not be stochastic. So it may be something that just happens quite often or all the time, which I think is important if you're trying to target these 2 antigens at the same time. I won't dwell on this too long. I think the short-term persistence of AUTO3 probably precludes it from being a good treatment for BALL. However, we have tried it out in diffuse large B-cell lymphoma, where CD19 negative escape is also a problem. And I think there the data are show that maybe it's a bit more of a useful treatment, particularly for patients where you can manufacture a decent dose of CAR T cells. But I think the conclusion of this is that it is feasible to co-express 2 CARs efficiently. We didn't see any particularly unexpected toxicity because of doing that. CD22 targeting is difficult at a preclinical and also a clinical level. And also I think this work emphasized the importance of having a very good manufacturing process and making sure you have long-term persistence. I'm going to come back to this a little bit later on, but I think just to let you know where this is going, I think what we've done is we've tweaked the co-stimulatory signals, and we've also added some genetically encodable modules that render this T cells resistant to TGF-beta and also PD-1 type signals. And actually, if you do that in vivo and in vitro experiments, at least, that looks quite promising. And this might be interesting to pursue, probably in DLBCL rather than BALL. So now I'm going to move on a little bit, and I'm going to talk about another BALL strategy. And I think here we kind of have to take a little bit of a step back. So I said, initially that there were 2 main challenges with BALL in terms of trying to, get as many patients to stay in remission as much as possible. The first challenge was CD19 negative escape, but the second challenge is long-term persistence. And I think that it's very clear in BALL studies with CD19 CAR that if your CAR T cells stop persisting relatively early, and it's difficult to know exactly what the magical time point is, but certainly within 6 months, it's almost inevitably your patient will end up recovering their normal B cells, having an MRD positive signal, and then having a frank relapse. At a year, that happens, more cases than not, and then maybe at 18 months or 2 years, maybe then you can start to relax. But it is really important in ALL, as opposed to DLBCL, that you have long-term persistence. And I think we decided to try and approach this problem really by some very elegant work from quite a long, from 20 years ago now on bispecific T-cell engagers, with the observation that if you tune the binding kinetic of the CD3 component of a bispecific T-cell engagers, you can actually end up with more inverted commas physiological signaling and less T-cell exhaustion. And we screened a whole load of different CD19 binders, and we found one called Cat19 that seemed to have a very interesting binding kinetic that also matched a very interesting as biological property when put into a car scaffold. I've just put the on and off rates here, just as an illustration. FMC63 is a binder used in nearly every single other CD19 CAR, with the exception of UCAR-T19, which uses 4G7. But FMC63 is used in Yescarta and Kymriah, in Lysocell, and pretty much everything. It's sort of the canonical CD19 binder. It's a very high affinity binder. Depending how you measure it, it's probably even sub-nanomolar. And as such, it has a very fast on rate and a very slow off rate. Cat19 has the same very fast on rate, but has a much faster off rate. So it grabs quickly and lets go quickly. And again, we published this a while ago, so this is relatively old data. So, you can look at Sarah Grashan's paper in Nature Medicine from a couple of years ago. And I think the in vitro consequences of this are you get equally good killing, but you also tend to get more proliferation and less exhaustion. And this carries out also in mouse studies. And I think the idea is that each interaction is more transient and less intensive. So what you would hope to see is less exhaustion, less apoptotic markers in the T cells, and also less cytokine release, and with that possibly, hopefully, less toxicity in the patients. So we tested this CAR. So this is just a plain CD19 CAR in a pediatric study. And again, we've published this a while back. This is an older study than the Auto3 study. So this was actually a simpler study in that it was a single fixed dose. Patients, again, were preconditioned with cyclophosphamide and fludarabine, and obviously this is an autologous CAR-T product. And importantly, this is a lentiviral product with a manufacturing process really geared towards persistence, for example, with no use of exogenous cytokines if at all possible during the expansion process, and as short an expansion process as possible. And I think this study was quite gratifying in the sense that we saw in patients really what we hope to see from preclinical data and sort of from the proof of concept. I think what we saw is very low toxicity. So on the bottom right, I've put the toxicity down. So Persis Amrolia, who was a PI of the study, didn't see any grade 3 or higher CRS, and which is quite remarkable actually in this kind of a setting when you'd expect to see quite a lot of grade 3 or 4 CRS. And we saw roughly the same CR rates and roughly the same sustained responses as with other well-engrafting CD19 CAR-Ts. The other thing that we saw that was really nice in this study was it was a pleasure to work with in terms of long-term persistence. So we could pick up CAR-T cells by flow in all patients, even many years out. So you do tend to get a very stable long-term persistence by flow and by PCR. So I think from that sense, I think we had a manufacturing platform here and a CAR that could give us very long-term persistence and also the added benefit of reduced toxicity. So I think what we did next is we said, well, rather than developing something new, oh yes, the adult hematologist. I have to mention this as an adult hematologist. Actually, although this may not interest many of you, the big problem in adult ALL, as well as the CD19 negative escape and persistence, is actually toxicity. So adults can tolerate immunotoxicity of CAR a lot less well than children, for probably reasons that you better understand than I do. So actually, probably this particular CAR, this Cat19 4-1BB zeta, is probably particularly well suited to adults with ALL, where a real barrier to treating patients there has been the severity of toxicity and how poorly tolerated CD19 CAR T cell treatments have been in that setting. And actually, we've just very recently, Claire Roddy has just recently published our findings from an adult study for the ALLCAR19 study, which again is a fairly straightforward autologous CD19 CAR T cell study with flu-cy conditioning because we were so worried about toxicity, we split the dose and also titrated it. So sort of belt and brace approach from the point of view of trying to reduce toxicity from a dosing regimen. This is bearing in mind that the data from our colleagues at the time of this study in Penn had showed quite a lot of grade 4s and also grade 5 CRSs. And I think this, so if you're interested, we published it in JCO just a couple of months ago. And again, we saw very, very similar results to the pediatric study. So really very nice long-term engraftment and very nice long-term persistence, very long-term B-cell aplasia that correlated with the long-term persistence, and also nice outcomes in terms of reasonable high response rates and a reasonable number of patients making it through to the 2-year mark. Interestingly, CD19 negative escape, although this has absolutely no statistical power because it's a very small number, it's only 20 patients, but CD19 negative escape seems to be a little bit less, although certainly occurred, but seemed to be a little bit less of a problem in adult ALL and tended to occur in younger patients. So with that in mind, we had a very nice platform of a CD19 CAR that was sort of proven in the pediatric and adult setting. And we decided then to have a second go really with CD22 targeting, building on what we've learned before. And I said that CD22 is a very difficult target. So we spent about 2 years trying to make the best possible CD22 CAR we could possibly make. I can't give you any sort of magical insights because I didn't find any. I think what we did was we vaccinated a lot of rats and hyperimmune mice with the proximal 4 domains of CD22, so the domains closest to the membrane, and we screened these binders by biophysics, by stability of the binder, and did the binder have a reasonable affinity and a high degree of specificity, and then the ones that sort of passed through that biophysical gate, then we went on to do functional screening in primary B cells against against the cell lines engineered to have very low copies of CD22. So we engineered a cell line to have 256 copies of CD22, which is which is well below 1,000, and purely by and we identified a couple of binders that performed really well. I haven't shown it here, but I think the most important thing is actually IL-2 release. If you can get IL-2 release to very low antigen density, that you're getting pretty decent signaling. And one of the binders, 9A8, was actually very good at getting IL-2 release at very low density. It was very specific and didn't give any background, any background signaling in the absence of antigen. So we took that forward. Now, we have a really good CD19 CAR in a lentiviral cassette, and now we've got a CD22 CAR. We don't want to make 2 products because that's too complicated. We don't want to make a TAN CAR. We could do another 2A peptide. And I think what we opted for here was really not to do any of those things, and to do something very simple, which is just do a double transduction. In essentially what you end up getting is a relatively complicated product, and I'm trying to illustrate it here on the bottom right, where you have T cells transduced with both vectors. So you have some cells that have the CD19 CAR and some cells that have a CD22 CAR, and then some that express both. And the fixed stoichiometry of a 2A peptide is now broken, so your product is much more complicated. There's advantages and disadvantages of these approaches. I think one advantage is we don't disturb the CD19 CAR. We know it's working. We completely leave it alone. We don't try and stick a 2A peptide in another a reading frame for another CAR that might affect its expression or vector performance. We completely leave that alone. Actually, breaking the stoichiometry is worth doing, because if, for example, the CD22 CAR fires too much and you get exhaustion, or there's some reason why targeting CD22 results in poor long-term persistence of CAR T cells, you still have your single positive, your CD19 CAR T cell population. So at least that will protect the patient from relapse. And from a translational medicine point of view, you can see the relative engraftment of these 2 subpopulations in the patients. From a regulatory perspective, it is more complicated because you've got more parameters to measure. But what I always answer when people worry about that is these products are so heterogeneous anyway. I mean, you can have one product that's predominantly CD4, one that's predominantly CD8, etc. So a bit more heterogeneity into the mix is maybe acceptable. Maybe we should embrace randomness with these products a little bit. So this is the approach that we took, and actually it works really nicely. So I'm just showing you a little bit of data here, where you have a single transduction with CD19 CAR, a single transduction with the CD22 CAR 9A8, and then double transduction. And you can see when you do a double transduction, you can see these nice 4 quadrants. And then, the double transduced cells do exactly what you expect them to do. They kill double or single positive target cells with really no effect compared to the single transduced cells. And they maintain their activity against ultra low density targets as well. When you put these mixes of cells or these double transduced products in mice, you get some interesting effects. Obviously, in a model, a NALM6 model with CD19 knockout, which is sort of a very crude model of CD19 negative escape, it works well. But the nice thing that you can see is that if you look at the populations that engraft, if you give these cells to mice who have NALM6 wild type, which are both 19 and 22 positive, or 19 negative, you can see different subpopulations in grafting. So if you give these cells to mice with CD19 negative leukemia cells, the CD19 single positive CAR T cells tend not to expand, which is what you'd expect. But the double positive ones do. Presumably, they're driven by the by the CD22 CAR. So so that's the idea. And this was plugged into the original very simple process that was geared to long-term persistence. And here's an example of a product, and I guess it's pretty much what you'd expect, where you've got 4 populations: negative, double positive, and single positive. In case anybody's interested, we've made really good anti-idiotypes for the 2 CARs, so that's why the staining looks so nice. And this is pretty much as far as I can take it, I'm afraid, until until kind of a bigger presentation. But the MHRA, which is our regulator, our FDA, has allowed us to amend the original CARPAL study so we can do a double transduced product, which is quite nice. We didn't have to start again from the beginning. We kept the dosing exactly the same. And this fax plot here is not from a mouse or from our product. It's actually from the peripheral blood of one of the patients treated on the 3rd cohort of the CARPAL study, which is this CAT19 9A8. And I guess, as they say, watch this space. Fingers crossed we get the long-term persistence that we did with the original product, and we see reduced CD19 negative escape. These are studies that do take a while to do, because obviously CD19 negative escape doesn't happen, immediately. So I think with that, then, that's sort of 2/3 of my talk done. So I'm half an hour, so that's pretty good. All right. And now I'm going to move on to our experience with GD2, and I'm going to really give you a little bit of an overview of a study that I did in collaboration with Karen Strathoff, that was funded by CRUK. And again, this is all published work. We published in Science Translational Medicine at the end of last year. Strathoff et al. If you want to look a little bit more. I don't need to tell this audience, what neuroblastoma is. I think the main reason that we went after it is not because of the unmet clinical need, which there clearly is, but I think the real thing is actually it's got a really good antigen. I think one of the emerging challenges in moving the success with CAR T from lymphoid malignancies and myeloma into solid tumors is actually finding good antigens and actually making sure that you can target these antigens safely. You can't obviously go after a tissue-specific antigen like CD19 if you're targeting an epithelial cancer, because you're going to end up hitting vital tissues. Neuroblastoma expresses the fetal ganglioside GD2 at very high density, and pretty much every single case of neuroblastoma expresses a lot of GD2. GD2 itself, postnatally, is restricted to very low-level expression on peripheral pain fibers and also brain parenchyma, with some increased density in some particular CNS loci, like the locus ambiguans. And obviously, many of you in the audience will know that there's a huge amount of experience targeting GD2 with therapeutic monoclonal antibodies. And there has been some early experience that Steve mentioned with first-generation chimeric antigen receptors targeting GD2. And I think in this study, what we wanted to do was we wanted to see, could second generation receptors, that is, receptors that transmit not just a killing signal, but a co-stimulatory signal. Could these be used safely in patients? Could we not end up having CNS or peripheral neurotoxicity? And also, could we see some signs of immunological activity of these T cells in these patients? And again, we're exploiting the fact that GD2 is about as good a solid tumor antigen as you can get. In terms of the design of the cassette, again, it's a bicistronic one, a bit like the AutoTree cassette with a 2A peptide that self-cleaves. And we have two components. One is something called RQR8, which is essentially two copies of the bit of CD20 that binds rituximab. That's probably the simplest I can put it. I've left out the reference there. If anybody wants to read more about it, it's Phillips et al. From Blood from about 5 years ago. And the idea of this is that if you run into trouble and you have to deplete the CAR T cells, you can give the patient a couple of doses of rituximab, which is relatively innocuous, and then hopefully that should deplete your T cells. So it's a suicide gene. The chimeric antigen receptor itself is based on an intermediate affinity GD2 binder that's been humanized called HUK666. The spacer is an FC spacer with FC receptor recognition sites mutated out. And it's a second generation receptor that transmits a 28 zeta. The reason we picked an intermediate affinity binder was this is a more potent receptor than had been tested before in this setting. We found that a receptor of intermediate affinity avoided recognition of very low antigen density, but recognized the high pathological antigen density very effectively. So we're hoping that this would discriminate between pathological and physiological expression of GD2. And the receptor itself is a sort of a souped up retroviral vector with a scaffold attachment region to homogenize and stabilize expression. This study was designed to be very, very cautious because of worries about toxicity. So the study initially started with a very low dose of CAR T cells with no prior lymphodepletion. And it was really only when we got to cohort 4 where we had a decent dose of CAR T cells. And there we, as well as fludarabine and cyclophosphamide. So in cohort 1, 2, and 3, which our regulator allowed us to do in the end with just one patient in each cohort, we didn't see anything. It was really only when we reached 1 × 10⁸ with precondition that we saw something happening. And I was just going to, because this is, I suppose, many of you are clinicians. I thought it'd be interesting to go through, our experience with one of these patients, which in many ways really resembled a lot what you might see in a patient with a bulky lymphoma after CD19 CAR T cell treatment. So this patient ended up getting spiking fevers and a drop in systolic blood pressure at around day 6, day 7. And this coincided with a very, very high peak of IL-6. In fact, we've had to do numerous dilutions, and I think it's well over 10,000 picograms per mil. And then this quickly resolved with some tocilizumab, which was very, very nice to see. The patient then recovered and was actually quite well after that. And then a little bit later, from around day 7 onwards, or probably around day 7 or 8, developed weight gain, ascites, and raised ferritin. And this really felt like a macrophage activation syndrome. And patient received supportive care and slowly resolved. And then there was sort of one last kind of interesting phenomenon in that the patient then later on developed back pain, a rise in LDH, potassium and phosphate. And had a high LDH, and this really looked biochemically like a tumor lysis syndrome, which was treated, just supportively with IV fluids and allopurinol. So what happened to this patient in terms of disease burden? So this is the bone marrow trephine left and right, top and bottom at day 0, and on the left. And as you can see, the patient's bone marrow is completely infiltrated with neuroblastoma cells. The architecture is completely effaced. And then at day 28, then this is the post marrow. You can see that it's clearly not normal, but there's very little neuroblastoma left. There's an arrow pointing to just one island there. So you can see that there's been a massive amount of necrosis actually. Which is interesting, and it makes it difficult to get any really good translational information from from these kind of biopsies when really all you have is necrotic tissue. But that's really what we saw. And then if you go on to look at the MIBG imaging, again, day 0 and day 28, anterior and posterior, you can see that there has been a reduction in the total burden of the disease when you look, for example, at the vertebra and the pelvis, but it's clearly not all gone. But I guess something has happened here. Another interesting thing that happened, that happens with these patients is actually a cytopenia, which again really resembles the cytopenia that we see after a CD19 CAR T cell therapy in pediatric BALL. If there's been a lot of inflammation, a lot of cytokine activity in the marrow. I think, sadly for this patient, then went on to develop clinical symptoms, and then re-imaging and remarrowing showed recurrence of disease at around day 45 in this patient, and then went on to die of pseudomonal septicemia. And if you go into Karen's paper, Stratoff et al., you'll see some clinical details about a couple of other patients. One other patient had a sort of a syndrome very similar to this, which with something that looked like CRS and a reduction in MIBG imaging. One other patient had a lot of localized supraclavicular disease that fluctuated and partially resolved and had necrosis and biopsy, but then relapse around the main original site of disease. But I think the conclusion from this really was I think you could take home a few messages from this. I think one thing is that you can use a second generation GD2 CAR and you can not get neurotoxicity. So very importantly, none of these patients developed any kind of peripheral neuropathy, any pain syndrome that you might see with a therapeutic antibody. And I think one patient had a headache for about 24 hours, a normal MRI scan, but we didn't see any other signs of CNS disease, and that was during a sort of a CRS type event. So I think targeting GD2 with this particular receptor, at least with a second generation receptor, I think it is safe from the on target off tumor perspective. So I think that's useful. I think the other thing that's interesting to see is that you can see the kind of immunological effects that you might expect to see with a CD19 CAR. So that's also gratifying to see. So there's no magical reason why you can't get some kind of immune activity going if you get the conditioning and the dosing and the manufacturing right. But clearly, there is still more work to be done. I think at this point, it's worth mentioning that there's some really encouraging data, with GD2. So I think the group in Rome from the Ospedale di Bambini de Gesù have done a way more GD2 CAR T patients than we have with a third generation 28-4BB Zeta CAR. And I hope to see their data published soon, because whenever they present it, there's more and more patients treated. And that looks quite similar to Karen's patients, with CRS type syndromes, partial reductions. And I think even in some patients with low disease burdens, they're seeing complete responses as well. So that really, I think, is even more encouraging. And I think then from Stanford, from Robbie Magner and et al., there's some very encouraging data that's been, very early data presented with the IPG, which is also GD2 positive, with some spinal disease and some central nervous system disease with partial responses as well. So I think GD2 is going to be, the next hottest target in CAR T after CD19 and BCMA, and there should be an awful lot more activity directed at it, because it looks like it's a really promising target. And obviously GD2 is expressed on neuroblastoma and diffuse midline gliomas, and also sarcomas, both osteo and Ewing. So it's a and then in adults, also in disease like melanoma and small cell lung cancer. So it's a target with a reasonable amount of potential. But obviously, I don't think anybody is seeing the kind of sustained CRs that we see in ALL or diffuse large B-cell lymphoma. So there's clearly more work to be done, although maybe we've got one good target we can play with now. And I think the problem is this, in a nutshell, which is really the tumor microenvironment. So in solid cancers, as well as being harder to target and harder to access, because they're not in places where T cells normally traffic, typically surround themselves with a particularly hostile microenvironment. It's not to say that B-cell malignancies don't, but the situation seems to be worse and more complicated in solid tumors. And, this is, I kind of like to tinker around with electronic things, and I think this is sort of the way I look at the solution, is that if you've got a problem, then I think you develop a toolkit of genetically encodable components to try and put them all together to try and end up with a T-cell treatment that overcomes all the different challenges. And I think it's important that this is done a bit like the CD19 or the BALL story, where you try and do things incrementally and learn as you go from clinical studies. So I'm just going to very briefly, in the last few minutes, tell you about our next generation GD2 CAR T-cell study, which we will hopefully start next year. And I think this is really, again, with the idea that you build on what you've done already. So we're leaving the GD2 CAR, the HUK 66628 zeta, and the safety switch RQR the same. So we're leaving those alone because clearly they seem to be doing what they need to be doing. And then we're adding 3 additional genetically encodable modules. One module called truncated SHP2 blocks PD-1 type ITIM inhibitory signals. A dominant negative TGF-beta receptor is a very old and well-described component that renders T cells resistant to TGF-beta. And then because I think these T cells do need to see, they don't see antigen. So unlike CD19, they can deplete quite quickly from the circulation. So I think we've given them a constitutively active cytokine signal to keep them going, particularly in the absence of any stimulation in circulation until they find antigen, and maybe to improve their performance then once they do find antigen. And then we're trying to get all of this together into one T cell. We couldn't easily fit it all into one vector, and I think because we have some courage now with doing double transductions in the ALL setting, I think we've taken a double transduction approach, which is there are probably technically more elegant ways of doing it, like directed insertion into 2 loci, etc. But but that's actually quite tricky to do in GMP. So we've gone for the very simple option of making 2 gamma retroviral vectors and doing a double transduction. The design is kind of a little bit interesting in that both cassettes have RQR8 and GD2, and then one of them has the CCR, which is quite big, and then the other one has truncated SHP2 and dominant negative TGF-beta receptor. And the nice thing about this is that even if the cell is single transduced, it will still have the CAR and the suicide gene, so you don't have to do any post-transduction sorting, which is always difficult to do during, it's never nice to try and sort cells after they've been transduced just before you cryopreserve them. And here's just an example of, some double transductions, and it's pretty much what you would expect. That if you do a double transduction, you tend to see 3 different populations, or 4 if you count the negatives. Interestingly, because in culture, these components actually do give the cells a bit of an advantage. There is a bit of TGF-beta in serum that we use to grow cells in, even AB serum, even human AB serum. And also the cytokine receptor also gives a slight proliferative advantage. So we do tend to see more double positive cells at the end of a manufacture than you would think than from CD19 and CD22, which is interesting. Anyway, let me talk a little bit about the different components. So a key inhibitory pathway is SHP1 and SHP2. So these are unusual SH2 proteins in that they're only ones that have a phosphatase. And what happens is when an inhibitory receptor that contains an ITIM, or immune tyrosine inhibitory domain, of which PD-1 is sort of the classic example, gets phosphorylated, SHP1 and SHP2 dock to the phosphorylated ITIMs and then go on to dephosphorylate the activatory motifs of immune receptors, which are called ITAMs, or immune tyrosine activating motifs. I know that's all a bit of a mouthful, but it's really a very nice sort of balanced, very nice balanced system, between ITIMs and ITAMs. And there's loads of inhibitory receptors that contain ITIMs. So PD-1 is just the best known example, but BTLA, for example, is also an important inhibitory receptor. And you can try and modulate these inhibitory signals in a few different ways. You can give systemic checkpoint blockade, for example. I think a problem with that is you might get toxicity outside of autoimmunity. You may not get very good penetration of the tumor, and also you're only blocking one inhibitory pathway. The other thing you can do is you can genomically edit PD-1 out. Again, it's only one pathway, and it's actually tricky you have to do an electroporation as well as transduction. So from a manufacturing perspective, it's quite difficult. So what we've done is something very simple. So SHP2 docks to the ITIMs through its SH2 domains and brings in this phosphatase. So what we've done is we've just chopped off the phosphatase and we just have the SH2 domains from SHP2. And what we think this will do is it just binds to phosphorylated ITIMs and then prevents the full-length natural SHP1 or SHP2 molecules from docking and thereby just stopping them from docking and inhibiting TCR or in our case a CAR. So and actually the SH2 domains are really small. They're smaller than GFP, for example. So it's very easy to overexpress them at high levels. Again, I don't have time to go into a huge amount of data, but I can just show you a little bit of data that if you have our original GD2 CAR T cells and you challenge them with a target cell that expresses GD2, they make lots and lots of interferon gamma. If you then challenge them with a target cell line that expresses PDL1, they make much less, or if you really push it, almost no interferon gamma. However, if you're co-expressing our truncated SHP2 module, you then end up restoring activity. So that's just a very simple illustration of what it does. TGF-beta is a fundamental inhibitory signal in the tumor microenvironment. So it's one of the most important inhibitory signals in the tumor microenvironment. And this is work that I've got no claim to. This is a component designed in Baylor, I think in the late 90s, that Kat Bollard, who when she was in the Center for Cell and Gene Therapy, really pioneered the use of. This is truncated TGF-beta receptor. TGF-beta receptor is a heterodimer, and it needs, and TGF-beta brings the two parts of the heterodimer together, and then you end up getting signaling and cross-phosphorylation and then SMAD signaling. If you overexpress one of the two chains and you truncate it so it doesn't have an endodomain anymore, obviously the two chains come together with TGF-beta, but then you no longer have the opportunity of cross-phosphorylation and no signaling. TGF-beta receptor is extremely potent receptor, but is expressed at very low level on T cells, even though it's profoundly inhibitory. So it's actually quite, although there's no reason why expressing a truncated TGF-beta receptor should be dominant negative from a sort of a molecular perspective, functionally it is, because you can overexpress it quite easily to such high amounts. You really get no TGF-beta signaling. And again, here's another very simple illustration, again with interferon gamma. If you have the regular GD2 CAR and you try and stimulate it with GD2 positive cells, in the presence of TGF-beta, you get a profound inhibition of everything from killing to proliferation to interferon gamma release. But if you co-express TGF-beta, you actually get enhanced activity. The interesting thing about this module, it generally makes CAR T cells work better everywhere, even in the absence of exogenously added TGF-beta. And I think, there probably is a little bit in serum when probably even in blood when in vivo as well. So that's two of the five components. And then the third one is a constitutively active cytokine receptor. And this is just really a component that just forces together the two parts of a cytokine receptor together to make the cell think that it's receiving a cytokine signal. And I'll go through this relatively quickly because I'm running a little bit out of time. And I think what it does is in vitro is it makes the cells turn over and not die in the absence of cytokine in culture. They eventually do in the absence of antigen, but it just keeps them ticking over for a little bit longer. And then if you do serial challenges, you get a better performance. So the T cells keep on proliferating despite, in this case, 7 or 8 or 9 serial challenges done twice a week. If you bring everything together and you do sort of a relatively crude model with a xenograft with CHLA-255, you get, I think, very, very convincing data showing that the enhanced cassette is much better than the original cassette. And I think this is an experiment done not by the pediatric team, by Fred Arke, also in our group, but with a small cell lung cancer xenograft. And I think he's had the patients to actually take apart each individual component and kind of show the sort of synergy you get from bringing everything together in terms of tumor growth and also and cytokine release within the animals, perhaps indicating that there's a balance to be met between efficacy and toxicity, which is always the case with these things. So this is really the next clinical study that we're doing in neuroblastoma, which is this multi-modular double transduced 5-component studies that hopefully will start early next year. And if it works out, we can extend it to other GD2 positive indications. And I think with that, I'm pretty much at the time when Steve told me to wrap up. So I'd like to just thank a few people. I'm Sean Cordoba, Simon Thomas, and Shimobi Noha did a lot of the binder engineering work. Eva Kokalaki did a lot of the nearly all of the CD22 CAR T work. But I would like to particularly thank 3 people because, as I said, I'm not a pediatrician. I'm an interloper to St. Jude's. So the only reason I'm able to do any of this work in pediatrics is because of 3 friends and colleagues who've worked with for many years: Persis Amrolia, Sarah Grashan, and Karen Strathoff, who all work at the Institute for Child Health and Great Ormond Street Hospital, which is part of the UCL campus. So with that, I will be quiet. Thank you very much for your attention.
Thank you, Martin, for your wonderful talk. Are there any? Probably I should give everyone time to enter their questions. There are a few already in the chat. Can you comment in regards to your, constitutively active cytokine receptors? Are you worried about toxicity, or have you seen any toxicity in mice?
Yes is the answer. So I think that if you give the most interesting observation we did is that we ended up seeing a lot of xeno-GvHD earlier than we thought. So that was sort of the first signal of toxicity. The second thing is, in a dose-dependent manner, if you give a lot of T cells, you can end up, getting mouse CRS with sort of weight loss and splenomegaly. So I think this is, I think this is always a challenge with CAR T, that you're always going on one hand without having enough potency and enough activity, and on the other hand having CRS and macrophage activation syndrome. I think one of the nice things about the which I didn't have time to really show with our constitutively active cytokine receptors, that you can actually chop bits off the 2 chains and you can sort of tune up and tune down potency. So that gives you sort of an extra possibility of tuning, which I didn't have time to go into. But I think this is one of these things we just have to see in clinic. The double transduction also gives you a little bit of freedom, in that you can, in a study, for example, remove the CCR component if you had to, or else maybe you could dial down the amount of vector you use to reduce the MFI and also the proportion of cells that express the CCR. So, I guess we'll see what we see in clinic, but there are a few options during the clinical study and maybe subsequently to tune it.
There's another question is, what are your thoughts on controlling cytokine release syndrome? In particular, since you're now going in with, CAR T cells, which have additional genetic modifications, what are kind of your thoughts?
Yes, we have a suicide gene, so I guess that's one thing. So I suppose if you really had to, you could give the patients rituximab, and that should hopefully deplete most of the cells. Obviously, then that probably renders the patients will inevitably relapse then once you've depleted the T cells. We have played around a lot with tunable chimeric antigen receptors, and we're about to publish a very simple paper in a tetracycline-induced protein disruption receptor, and how quite a few have been described. I have to say I've kind of been less enamored with them. In our hands, at least, it's very difficult to maintain the signaling of a monolithic or classical receptor when you start messing with it to make it tunable. And as well as that, I think pharmacologically, there's so many options. There's so many things you can do without having to resort to engineering, whether it's just dexamethasone or tocilizumab or IL-1 blockade, or even other things that can inhibit T-cell signaling. So I'm not a huge fan of tunable chimeric antigen receptors. I may eat my words in a few years' time, but at the moment I haven't decided to incorporate them into this generation of studies.
There's one question, I don't know if you can answer that, in regards to your current bispecific study. Can you comment what happens to individual CAR T population over time?
I think we have a preclinical poster at ASH. And I think there may be a little bit of data you can look at then, but Persis will kill me if I spill any beans, so I have to behave myself.
All right. So we must have some listeners from the UK, because there's one question: when will be, your studies open for targeting diffuse midline gliomas with GD2 CAR Ts in the UK?
Yeah, I mean that's a hard question to answer. I mean, we did put in a grant to our medical research council, if any of them are listening, and they rejected it. So unfortunately, that meant that actually we couldn't do a GD2 study, and Stanford got there first, which is, that's the way these things go, I guess, with grant reviews. I think we are trying to, Karen is trying, and I don't want to put her under any pressure, but she is trying to, we have some vector left from the original, neuroblastoma study, and she is trying to, use that to do a simple, study, which is the plain vector in DMG, because the Stanford's group data has been so promising to see what would happen with our receptor. But I cannot make, any promises. That's dependent on external funding and on other things. But, we do a vector, so we could do it if, if we had enough support.
There's one question in regards to your SHP2 kind of inhibitor module. Why doesn't it affect the CAR T cell receptor itself?
It doesn't interact with the CAR T cells. So you have ITAMs, which are the activating, activating motifs, and ITIMs. So SHP2 binds to the phospho-ITIMs, so it doesn't really interact directly with the chimeric antigen receptor. So there's no real reason why it should inhibit, or have any real effects on the CAR. I mean, maybe I didn't make that clear. So it binds the ITIMs. And stops full-length SHP1 and SHP2 from docking, which would then go on and dephosphorylate the ITAM, which is the bit that's stuck on the chimeric antigen receptor.
There's one more question in regards to the GD2 CAR study you presented. In the patients where you had, tumor lysis or CRS, did you see any evidence in the end of antigen spreading or, responses on, which would be indicative, which are, endogenous, let's say neuroblastoma specific?
No. We didn't look very hard, but we didn't really see any sign of that. I think it really looked like all the activity was really driven by the GD2, which is maybe not so surprising. I mean, I think most pediatric solid tumors are cold, aren't they? They're not hypermutated and they're not immunologically that activatory.
One question, I know you have a safety switch, but as you kind of put in more and more things into these cells, do we need in the end robust preclinical assays to look at malignant transformation or the potential? What are kind of your thoughts in that regard?
Yeah, I mean, I think you have to, have a balance. I think, I think, people like Steve and others who are experienced in the field, will know that often clinical findings are really totally surprising. I think the clinical findings with experience in ALL, for example, of CRS, neurotoxicity, CD19 negative escape, etc., were completely not predicted, from preclinical data. So you have to be temperate and not be. If you're too cautious, you never do anything. I mean, I think what we've done is we've just done some very long-term cultures where we put the cells both into mice and in ex vivo culture and just left them alone and see what happens. And I don't, the CCRs don't immortalize the T cells from what we can see. But I think it's important to kind of keep a certain pace of clinical development. Otherwise, you'll never do anything.
I completely agree. So there right now no additional question, and we are at the top of the hour. So I would, Martin will graciously agree to stay on if there are additional questions, but I would formally just close the session and remind everyone that next week, Dr. Kirsten Nies will talk from St. Jude on interventions to prevent the remediate frailty in childhood cancer survivors. So, I would like to thank everyone, and I'm just looking through the chat if I kind of missed any major questions. There's one question I think is probably driven by, you probably are aware that there are at least 2 recent studies where at least some reports about genotoxicity of, genetically modifying what are kind of your thoughts, in your program, since you have a lot of also experience? What are kind of your thoughts overall on that?
Yeah, I think that's a good question. I think there's two reports. One was a study in Australia, which used transposase and naked plasmid DNA to manipulate the T cells. That's a pretty unusual thing to do. So there's many hundreds of patients worth of experience with gamma retroviral vectors and lentiviral vectors, with really no evidence of transformation of any significance. So this was quite an unusual protocol. They were getting very high insertion copies in one of the cases, 50 or even 80, depending how you measure it. And it was two patients in one very small study. So I think in that case it's very clearly related to the platform. I don't think you can really extrapolate any of that data into the wider sort of, lenti and gamma retro CAR T cell world. I think you just have to say that's just a, strange, aberration. I think what they say in their discussion is that maybe they had too much transposase and it was just going around sort of chopping up and making double-stranded DNA breaks and didn't have enough plasmid DNA to sort of chew on instead. So I don't think that's any bearing on, unless you're in the unhappy situation of deciding to use a transposon for your CAR T cell program, in which case that would be a problem and you really need to do some thinking about it. I think the other one is really not a publication. I think it's a press release from Allogene, and that's related to the UCAR-T program, which actually I was involved in a long time ago. And I think the idea behind that study is that you make allogeneic T cells, you do two cuts, one in the TCR alpha locus to disrupt the T cell receptor to prevent GVHD, and another cut in the CD52 locus where you disrupt CD52 so you can then give the patients flu-cy and alemtuzumab, and that's to prevent rejection of the allogeneic, completely mismatched CAR T cells. And I think what's been found looks like a translocation, presumably between the two loci, although I don't think it's been said, I think that would be a reasonable assumption, between the TRAC and the CD52 locus. And I mean, in that one, I think if you go back to the original paper that we published with the UCAR-T19 approach, I think it's figure 6, with the last figures, there is a little bit of data, and you do get a reasonably high level of translocation, just under 1%. And it is two dead loci, so it's probably not of any consequence. And without actually knowing any more about it, you do wonder, did somebody do a fish find this translocation, and maybe it's maybe expected because it's in the product. If it's a very rare detection, if it's something that's obviously increasing serially in the patient, that might, suggest an autonomous proliferation, which might be more worrying. But my guess would be that it's just in the product and it probably is not of any consequence. But obviously it's a scary finding from a from a regulatory perspective.
Then there's one more question. Beyond GD2, what are you thinking about targeting in solid tumors? What are kind of...
I think in the pediatric setting, I think B7H3 looks like it's I would say it's probably the next target really to be explored. And there are quite a few studies, B7H3 open or opening. I think that looks like a good target also in DIPG and also in quite a wide variety of pediatric tumors. So I think, in the pediatric setting, I think that's probably the next thing to go after. In the adult setting, I think, and also possibly in the pediatric setting as well, I think IL-13 receptor alpha 2 looks reasonably promising in terms of CNS diseases. So I would probably, put those B7H3 and IL-13 receptor alpha 2 sort of next on the list. After that, it starts to get quite tricky, and you may have to move into more exotic targeting platforms, such as gated platforms and the like.
So that is actually funny that you mentioned that, because there was one question came in regards to the synod, so in regards to gated approaches. What are kind of your thoughts, kind of in general?
Yeah, I think the approach, one approach that's been published with IL-13 receptor alpha, with EGFRV3 and IL-13 receptor alpha 2, I think is very elegant. I think that's sort of the nicest approach I've seen. EGFRV3 is a very specific target for glioma, but is heterogeneous and tends to be patchy and easily downregulated, while IL-13 receptor alpha 2, you could worry that there might be some areas of on-target off-tumor toxicity. So I think the idea is you switch on, the EGFRV3 switches on the IL-13 receptor alpha 2 CAR, because the way SynNotch works, the expression doesn't, it hangs around for a bit, decays slowly. So even if not every single target cell is EGFR V3 positive, it's still enough in the milieu to express a second CAR. But then if the T cell wanders off somewhere, you'd hope that then that you'd lose the second CAR expression. I think that's a very nice and very elegant approach. So I think that's one reasonable, probably the most reasonable approach I've seen. I think otherwise it's quite tricky. I think if you have AND NOT gates where you have an inhibitory signal on the second CAR, the dynamic range of these systems is quite narrow. So you have to be very, very careful because it's quite easy to break them if you have overexpression of the first antigen. For example. I think it's definitely an advantage that engineered T cells have over, bispecific T cell engagers or therapeutic antibodies, but the technology is tricky. It'll probably take a next round of engineering to get it good enough, I think, for clinical application.
All right. So there are no more questions. Can I just have one last since you also, as you mentioned, work kind of with off-the-shelf products at some stage. Program is what you're pursuing is very much focused on autologous. Can you just what are your thoughts about the prospect of allogeneic products, in general?
Yeah. I think I get asked that a lot, and I think the answer is relatively complicated. I think you have to imagine it as sort of a balance. There's no sort of one magic answer. I think there's pros and cons. I think the advantages on the allogeneic side are obviously you don't have any vein to vein time. You're not relying on possibly poor quality T cells from the patient. So those are all two very good advantages. However, you do end up with GVHD, a fairly big problem you have to solve, and also the lack of long-term engraftment due to alloreactivity, which is very profound. And then on the other side, with autologous manufacturing, obviously the problems of manufacturing are not insignificant. Having said that, there's a huge amounts of engineering, and I don't mean DNA or protein engineering. I mean tubes and pumps and electronics and engineering effort going into autologous manufacturing. So the Miltenyi Prodigy, for example, was revolutionary. But there's been, there's other companies developing other products. And what this lets you do is reduce the cost and the clean room need for autologous manufacturing. And then, improved manufacturing processes are now down to a couple of days with a few days release, which sort of, again, shifts the advantage of that vein to vein time. And other stuff you can do with the autologous process, to reboot your T cells. So even if you've got a fairly bad T cell product, you can improve it. Initially it was all autologous was the only way to do it. And then it really looked like there was a huge amount of hype around allogeneic. And then I think you're kind of seeing some of the data emerging. People are talking about, we have to give multiple doses, which, takes away a lot of the advantages. And I think at the moment, my feeling is that swinging back to autologous with short, quick manufacturing processes and long-term persistence. But no doubt will swing back again in a few years' time, probably with iPSC-derived or, semi-immortalized T cells. So if anybody's certain that one platform is better than the other, they probably haven't thought enough about it. It's a bit more complicated. I think at the moment in ALL, again, if you look at the published, the spectrum of published data, for your patient, you'd want to give them back their own cells at the moment. Then if you can't, then you get an allogeneic. I don't think that's a controversial thing to say based on the data.
All right. Thank you again, Martin. I really enjoyed your talk and your conversation. And I would also like to thank all the audience who stayed on. And it was great at least connecting via WebEx, and send my regards to Karen.
All right. It's a pleasure. Thanks, Steve.
Bye.